Thank you guys for inviting me. It's a real pleasure to be here. And can you all hear me just fine? Okay, good. So as Marcel already mentioned, I did my PhD at Max Planck, but because Max Planck is not a degree-conferring institution, I actually had my promotion at Leiden. So when I did my promotion, it was the first time I'd ever visited Leiden. And actually, Hanno, a bit closer? And actually, Hanno is on my promotions committee, so it's a little bit of a homecoming right now. I'm going to present to you something maybe slightly different than the nature of the talks in this conference. I think for the most part you're getting a lot of synthesis of a lot of information, but what I'm going to do is provide you with the upfront kind of context, and then show you some of the data and some of the projects that I've been doing, and then allow you to sort of take away your own conclusions, and I'll have a takeaway message at the end. But I'm going to provide you with some of my, my actual core research. This nice introduction pretty much already filled you in with everything about me, but I have my PhD in archaeology and anthropology. I worked with the Plant Foods Group at Max Planck Institute, and my specialty has been diet and human evolution. And my main interests are in the encephalization of the human brain, so how humans acquired a large brain over evolutionary time. And I've also been doing work in the microbiome. So as part of my PhD, I actually did the microbiome, or characterized the microbiome of Hodza hunter-gatherers. And this also led into the work that I've been doing with how wild foods, particularly wild tubers, are influencing the microbiome, and in reverse. So what I really want to talk to you today about is this wild foods versus domesticated foods. So the majority of our foods are all domesticated cultivars, and this is an extremely big difference between cultivars and domesticates. And so, cultivars and wild foods. So wild foods, if you want an official definition, there's no intentional cultivation, no genetic modification, no changing of the habitat required of the thriving of the species, the setting in which it grows, and its natural predators. Whereas in domestication process, there's artificial selection, changing of phenotypic traits and often behaviors, both plant and animal, to facilitate the survival of that organism in tandem with humans. And human management allows us to select the kinds of traits that we're most interested in promoting in this species, rather than the kinds of traits that allow this species to thrive under natural selection. And so this really plays out into the kind of properties that we would expect, both physical and chemical, and we can segregate these two kinds of food groups. So wild foods tend to be higher in nutritional content. They have a lot of defenses for consumers, both physical and chemical. They have more complex structural elements. You can think of this as mainly fiber, but also the cortical tissue, the tissue around the outside of the food, I'm talking about plant foods, obviously, is much more difficult to actually break through and access the interior part of the food that may be more nutritious and edible. They tend to have secondary metabolites, meaning compounds that interact with different chemicals to confer the plant a defense mechanism, and actually can be toxic to a consumer, and this reduces the palatability of the food item. And they tend to be lower in caloric content. They haven't been selected for in easily digestible macronutrient elements that we as consumers favor in our foods today. Whereas domesticated foods are pretty much the opposite of everything I just described about wild foods. They have high caloric content typically. They have low chemical defense. If there are any kind of chemicals or toxins, they can easily be neutralized through thermal treatment, such as cooking. They're very palatable. They're very easy to consume and digest. They tend to be larger in size. we've selected for foods to be larger so we get more out of them, but they also lose a lot of their nutritional content. They're not as concentrated in micronutrient elements. And they've lost this kind of, these bitter, often toxic, but can also be beneficial compounds that make them that make wild foods quite nutritional. And this is termed the domestication syndrome. So cultivation is not just a sort of one-time innovation. This happened successively over maybe generations, thousands, could be even tens of thousands of years, beginning with early forms of crop, or actually before crops, of early forms of plant management. And this can be thought of, this is termed para-cultivation, so this is somewhat of a cultivated kind of behavior, but it's really just management of wild resources. So a nice example of this are the Baka foragers in the Central African Republic who manage the wild yams that live in the forest. And so what they do is after they've harvested some yam crops, they actually will save some of the yam tubers so that the plant can grow again and that they can go and harvest it at a later date. So this is management or consideration of the plant's needs and its productive viability in the future. But it's not strictly selecting or domesticating or even cultivating. So this is paracultivation. And we imagine that this probably has been going on for thousands, if not tens of thousands of years. And this is a sort of pan-human trait. This is something that is in our behavioral repertoire across all populations across the world. And from there, you move up in the scale of complexity, going to horticulture, which would be kind of analogous to farming, or gardening and cultivation of plants. And then you move up into the industrial scale of agriculture, so small-scale farming. This is usually non-mechanized farming, and you're normally operating on a subsistence basis, so what your family needs, and then on up to industrial-scale farming. And the important takeaway from these levels of cultivation is that you're moving in a scale of complexity from high complexity of the plant food, low complexity of the technology, to low complexity of the plant food, high complexity of the technology. And so as Dorian Fuller said, it's not a rare discovery, but this probably, the development of cultivation in agriculture had its foundations in an economic need rather than any sort of one-time innovation at one point in the transition from the Paleolithic to the Neolithic. And so this probably set up as waves of people and ideas and migrations happened, and that the need became more and more that people needed to produce foods and compete with other populations, or other populations wiped out certain people. And so it was really just a strategy that may have been known about for a very long time by everybody, but was really only adopted when the need arose. And archaeological evidence shows that the archaeobotanical remains that we find point to the Levant as being the origin of the earliest cultivated cereals. But cultivation happened at independent time points across the globe for different kinds of cereals and crops, going from the Levant to China to India, and then finally into West Africa. Okay. And the genomic evidence is also starting to really inform us about population migration and movement of people and early populations in Europe from farmers that arose in the Levant. And so this is just a map of a bunch of ancient human samples that were sequenced in this paper in 2016. And this is just a map of where all of the samples come from. So you have mainland Europe, and then you have the Near East, and then you have the Siberian and Eurasian steppe. And these are all full sequence ancient genomes. And then the data deriving from that is a SNP panel looking at single nucleotide polymorphisms to look at the differences between these two between all of these populations. And what the data are showing is that the initial rise of farming populations in the Levant were originally hunter-gatherers, and they are originally very genetically distinct from each other. And so this is demonstrating that it was really the movement of ideas rather than people. And then as these people began to intermix in the Levant, they also spread their ideas west and east and north and south. And so you had waves of people and waves of ideas coming in to eventually influence the other endemic hunter-gatherer populations in Europe. And so there's a mix of hunting and gathering genotypes, and a mix of early farming genotypes, and a mix of early Eurasian steppe genotypes that all have come together into what is now present-day Europe and European genetic differentiation. Sorry, I didn't really explain this figure to you, but this is a PCA plot. It's just a demonstration of how all of these different genomes are related to each other. And so what I've circled here are the different hunter-gatherer population genomes. And you can see that they fall outside of the main clusters of everything else, which are the farming populations. And then the gray dots behind all of the small colored samples is modern human genetic variation. Okay, so I'm going to just move right into what is hunting and gathering, and what is the definition of a hunter-gatherer. I'm not sure if anybody's really thought about what, a specific definition, how to classify hunting and gathering. And so there are basically two kind of definitions of this. One is how food is attained. So James Woodburn said that societies that obtain food and resources from purely natural resources, this is a hunter-gatherer. And other definitions would talk about the actual activity of humans in relation to their food. So no deliberate alteration of the gene pool of exploited species. So people that exploit purely wild foods would be hunter-gatherers. So in 1968, at the Pinnacle Man-Hunter Conference, it was estimated that 0.001% of the world's population were 100% foraging or hunter-gatherer populations. And in the 1990s, this had to be redefined to say that only 85 to 90% of the food must be wild resources, because it was realized that nobody was 100% hunter-gatherer. By 2014, no population in the world really currently meets any of these criteria for a hunting and gathering population. So we've lost our modern hunter-gatherers, unfortunately, and so we've really lost this information to ethnographic accounts. And so these become extremely important for us to really understand what is the behavioral type of humans that live in these pure wild environments, and what is required of people in order to subsist a pure hunting and gathering subsistence. So tubers have become sort of my specialty topic, and so what I studied were the wild tubers consumed. Tubers are actually extremely important in hunter-gatherer diet. They are extremely important in ancient human diet, and this doesn't get a lot of talking points because it's very hard to recapture what the archaeological record may say about tubers or plant foods for that matter. So that was a major goal of the group that I joined for my PhD, was to try and recover and look at the plant record and develop different methods to reconstruct the plant record of hunter-gatherers. And tubers really come into play because they're incredibly abundant. They're available year-round. All populations, whether they're tropical, neotropical, temperate populations, foraging populations, rely on tubers. And it's thought that they are a fallback food, which is not to say that they are only consumed when there's nothing else, but they're consumed daily as a daily component of the diet because they're always there, because you can always rely on tubers. You can't always rely on a large game kill or honey cache to be found or berries to be in season, but tubers are always there. And importantly, they are actually most nutritious in the wintertime if there is seasonality, because this is when the plant actually needs to store all of its nutrients inside the tuber. And so that's what the plant is actually living off of during the wintertime. So they become extremely important during winter when perhaps game become very lean and other types of plant foods are not so available. And tubers are largely foraged by women. And so we really get into the role of women in foraging societies when we talk about tubers. I just wanted to point out to you that probably the main, all of the tubers that you've consumed in your lifetime come from just a few regions around the world, and particularly in the Americas, where we have the native location of the origin of sweet potato, white potatoes, and cassava. And so these are all central to South American cultivars. And none of these are actually placed in regions where probably modern day people or you and me are coming from. And yet we consume these as probably a large portion of their diet. And then you have yams from Central Africa and the aroids such as taro coming from Southeast Asia. So there's a really limited range of dispersal of where domestic cultivated species of tubers come from. And compared to wild, this is maybe a bit cheeky, but this is just to say that wild tubers are everywhere. And so if you want to think about how or why tubers were exploited, well, it's because anywhere populations migrated, they knew that they could find tubers. They could probably go and dig somewhere where they recognized the plant type, the above ground plant type, and knew that they had a food resource. So tubers are incredibly important. It's estimated for one species of an East African tuber, there's approximately 80 tons of tuber per hectare for one single species. And for the Hodza in particular, they target on a regular basis roughly 4 to 5 different species, and this is only a subset of the entire set of species that they target. So you can see that they have quite a wide distribution to choose from. So just to introduce the Hodza to you, they are a tropical foraging population. Up until about 2014, they were probably our last hunting and gathering population that we really had regular contact with. There's probably a few other societies or small groups. You might not even be able to call them populations in the Amazonian rainforest and on the Andaman Islands, but really for a thriving population, the Hodza were probably our last. They've been studied for a very long time, beginning really with James Woodburn back in the 1960s, and then you had successive anthropologists such as my mentors, Frank Marlowe and Alyssa Crittenden, who have been long-standing Hadza anthropologists and ethnographers, and have been really formative in the body of work and research that we have on Hadza. So they forage in a savanna mosaic environment in eastern Africa and Tanzania near Lake Turkana. They really utilize a low complexity toolkit. They don't really have a need for fancy weaponry. They have bows and arrows, and they use a poison that they derive from a plant species. But other than that, it's basically a Schulian level tool technology for hand axes, for stone tool for pounding. So everything that they're able to derive from the environment really comes in a whole kind of natural unprocessed format. Plants comprise roughly 50 to 70% of total kilocalories in their diet, depending on season. And as I said before, tubers are a consistent resource. You can see from the figure on your right that the what I have circled is the tuber contribution to the diet by month. And other than 1 month, other than in July, when they're really able to forage for berries that have come into season, tubers sort of sit at this consistent, roughly 30% contribution to the diet year-round. So when I visited the Hodza back in 2013, what I was really interested in was following the women and looking at what they're doing, looking at tuber foraging. So I'm just going to kind of walk you through maybe a day in the life of a Hodza woman. She starts out in the morning sharpening her digging stick, and then they all gather and get together. It's women from young mothers to grandmothers, and sometimes small children, and often infants that are being carried along. And then they set off into the bush, and they walk for maybe 20 to 30 minutes. They find a tree that they know has different tuber species that are associated with it, and so they tap around with their stick and they listen for hollow points in the ground. And when they hear a hollow point, they know that there's probably some tubers underneath the ground, so then they'll start to dig. And they dig and dig for about 10, 15 minutes. It's fairly vigorous, and they create about a meter to 3 meter hole, and then they pull out the tubers, and then they continue. And after about an hour of this, they get tired, so they'll sit and they'll crack one open and have a snack and socialize, and the children will go play. So it's a fairly leisurely daily activity. It's part of their everyday ritual. So as the women eventually load up their sacks with tubers, they'll bring them back into camp. And usually in camp is when they'll actually roast the tubers. They occasionally roast them when they're actually out in the bush, but they'll eat them raw as well. But when they come back to camp, they always roast them on the main campfire, and this is when they share them with everybody else. So roasting and eating tubers becomes a communal activity. This happens 2 times a day, late morning and then in the afternoon. So there's 2 foraging bouts. And so actually the men will join to eat tubers when they come back to camp, but men otherwise are not eating tubers out in the bush. This is really more of a woman's main portion of her diet will come from tubers. As I said before, the Hodza target several species. These are 4 species that they target in particular, and these were the species that I was able to forage along with them and that were available at the time when I visited. These are the Hodza names, and they're only really recently taxonomically identified, and their Linnaean taxonomy is a bit uncertain and often changing, so we continue to refer to them by their Hodza name. You have Equa, Makalitako, Shimuko, and Panjuko. And these tubers are extremely fibrous. They're not potatoes, they're not sweet potatoes. They're more like, celery and sugarcane. Their favorite one is Equa, which is this long root that you see on the top, split open right here. It's actually more like a woody kind of tree limb that once you peel it, you can chew it a little bit and you just try and suck out as much juice. And then the picture up here on the top right is actually 2 slices of Equa, one pre-chewed and one post-chewed by me. And there's a lot of fiber you can see that's left over, and this fiber is not consumable. That you actually have to spit it out. So there's not a lot of food mass that they're actually consuming with Equa in particular, and even the other species have fiber that must be expelled as well. This is called a quid, so what's actually expectorated after chewing and sucking on all of the juice. And you can find quids, you can find Hodza camps by finding all of the quids that are being left over in their camp. There's only one type of tuber that does not have fiber, and that's this Panjuko. And Panjuko is really interesting because it's actually the same genus as sweet potato. So studies that look at trying to reconstruct diet and look at kind of properties of cooked foods using sweet potato and some other kind of meat, well, that's why they're using sweet potato, is because of its relation to the Hodza tuber. So this led me to question how digestible actually are Hodza tubers. If they have all of this fiber that has to be spit out, what's left over and how digestible is that? What does it contain? Is it starchy and does it have a lot of calories? So we know that humans have a very simple, non-specialized gut, and the small intestine has been increased relative to the colon over evolutionary time, also compared to other great apes. But we are still descended from, we have an herbivorous past. We're descended from herbivores. And we just have a derived gut morphology for very high quality, easy to digest foods. So this is kind of a paradox. How are Hodza eating these extremely fibrous tubers, but yet with a human gut? So just to kind of give you a little quick overview or reminder of primate evolutionary history, we have the ancestral primate form roughly 65 million years ago, which was probably an insectivore or a frugivore, so eating insects and fruit. And then you jump to roughly the Miocene apes 25 million years ago, and this would be really full-time folivore and frugivores. Then you have Sahelanthropus at 7 million, which is the species or the fossil that we think may be the earliest ape that split from all of the other ancestral apes and led to the line that we call the hominines. And then you have Australopithecus, which then changes. Oh my goodness, I am running out of time. Okay, then you have Australopithecines, and then you have Homo that really changed the diet strategy to different types of foraging, omnivory, and food processing. And so I wanted to look at in vitro or model digestion, so using in vitro digestion. And so rather than talking about foods as composition, such as fat, carbohydrates, and proteins, we wanted to know what is the bioaccessible fraction. So once you take a bite of something, it goes through your stomach, into your intestines. Some of it is absorbed and goes into your circulation, and then the rest is waste. And so this is the fraction of nutrition reaching circulation is the bioaccessible fraction, and this would be what we're really interested in learning about in terms of the tubers that are very difficult to digest. So what is bioaccessible? So this required that I actually dig up tubers. I pre-process them, so I roast them very briefly, just as the Hadza do. You have to peel them, you have to take all the fiber out, you have to simulate chewing. So this is kind of a process. And then finally, I get to what a lot of my friends like to call the robo-gut, which is where I worked in TNO and Zeist, and this is the TIM1. This is a fully dynamic model of the stomach and the small intestine. I don't really have time to go through it, but what I saw in the end was that there's a large variability in digestibility of the tubers, and they're not that digestible. They're maybe at the most roughly 60%, but 50 to 60% digestible relative to inputs. So that's not a lot of calories, and it's not being augmented. It's not being helped by roasting them. So this kind of fits into this picture of high variability of wild foods and also low digestibility, low caloric return of wild foods. So the conclusion is that cooking, the roasting, what we think of as being really important for our diet, making our foods more palatable, it's not really having an impact on the caloric return of wild tubers. And this suggests that cooking has a different role, is facilitating something else that is necessary, and that role is likely to allow access to the tuber because, like I said, it has a lot of these defense mechanisms, and one of them is a thick cortical tissue on the outside that must be peeled first. It's very bark-like. So in order to get into and actually access the tuber and be able to even chew it, give it a little bit more kind of pliability, roasting is thought to really facilitate the change in the physical property, not so much the chemical property, not really changing the starch, not gelatinizing, making starch more accessible, because that's not really relevant. They're not very caloric anyways. Behavioral patterns really matched the way the Hadza were roasting or not roasting. I don't really have time to go into this, but that's really my takeaway. And this is consistent with other ethnoarchaeological work looking at other wild foods that show that mechanical processing is what is really necessary in order to break into resistant foods, in order to really break open the cellular structure and allow starches that are inside the cell to be released. Thermal processing and cooking are not so important in this. So I'm going to move now into the microbiome work that I did, as well as collecting wild tubers. I collected fecal samples from the Hodza. This allowed us to publish a series of papers on the characterization, the taxonomy, the metagenome, the metabolome, the enterocyte inherent fraction of the microbiome, and still work that I'm in prep with the activity of the microbiome in relation to tuber digestion. So just ask for the next couple of slides not to take pictures and tweet, because this is data that are in prep right now and will soon be submitted for publication. So again, I'm simulating digestion, but this time it's fermentation. So first I had to pre-digest all of the tubers, doing the same process, and then filter out all of the metabolites that would have been absorbed in the small intestine, and then create the portion that would have gone into the large intestine to then be fermented in the colon. This was done again at TNO and Zeist. And here I'm comparing the activity of microbiome coming from fecal samples from Hodza and fecal samples from a standard Western microbiome, the lab technicians at TNO and Zeist. And also, this was tested against 9 different foods, 3 of them being the Hodza tubers, and then standard Western, so sweet potato, cornstarch, and then a control. This is what the TIM2, the in vitro colon, looks like. It's kind of TIM1's little brother. And I won't go into how it all works, but it's again a dynamic system that allows you to feed whatever substrate that you want to feed it. You can inoculate it with a microbiome of choice coming directly from fecal sample, and then you can take samples of the metabolites that are produced over a period of time. In this case, it was 72 hours or 3 days, and look at the activity, and also look at the structure of the microbiome itself. And this was very high throughput, much more so than TIM1. So we had 10 machines running in parallel for 10 different types of foods. Okay, so looking at the activity, what I really want to draw your attention to here is that in the baseline activity, so after an adaptation period, the Hodza microbiome, which is these orange bars, are overwhelmingly predisposed to producing lactate in response to carbohydrate than the standard Western inocula. And so this lactate is quite interesting. It's interconvertible to other different fatty acids, so acetate and propionate especially. It's thought to be incomplete fermentation, or fermentation that happens very rapidly, so as a result of a lot of input of carbohydrate. And it's also accessible to the human host. So we can actually absorb lactate through our colon, and this can actually be nutritionally relevant for humans. So in looking at the PC plot, you also see that these Hodza samples right here, this is at the first time point. So they're all enriched in lactate relative to all the other samples and relative to the standards at the same time point. And the distribution of the taxa and the microbiome are also responding similarly. So we're seeing an enrichment in different taxa that are lactate producers as well. So this is showing a mirroring of microbiome structure as well as activity, as we would like to expect. And also interestingly, looking at my previous results with the TIM1, the digestion results, the amount of glucose that's absorbed was actually inversely related to the amount of short-chain fatty acids that were produced. So in the Panjuco, in the green, there was very low glucose absorption, but in contrast, in the TIM2, it produced high amounts of short-chain fatty acids. So this is interesting to see. What we'd like to see is that the more nutrition that you can absorb in your small intestine, the less that there is to be produced in the large intestine. and then the converse of that. So these makalitako and shimuko that have very low amounts of starch, very low calorie, were really highly or easily absorbable in the small intestine and not overloading the small intestine, and then when they went into the large intestine, there's really not a lot of substrate to produce calorific nutrients. And also, very interestingly, I think probably one of the most important results that I'm seeing from this is that Hodza tubers, compared to other cultivars and the controls, are facilitating the enrichment and diversity of the gut microbiome. So starting from the same baseline, you see that over time, Hodza tubers are are really facilitating the number of taxonomic units that we see being produced in this community over time. So this is really supporting this hypothesis that our foods are not very conducive to allowing a diverse microbiome, which in a diverse microbiome is something that we think of as being very healthful now that we have a lot of research on microbiome. So just the conclusions and the takeaway from this. There are different metabolic profiles deriving from seen in Hodza versus the Western standard microbiome. And these may lead to different metabolic health conferring or nutritionally conferring changes in our own physiology. The wild tubers support a diverse microbiome well beyond the domestic tubers. And in sum, I think this really tells us that it's important to activate the entire digestive tract, not just small intestine digestion, but our colon is also incredibly important for our health, and the fermentation that goes on there is incredibly important for our health. But the foods that we eat don't necessarily always activate the entire digestive tract. So these are my last couple of slides. I just want to show you just a quick kind of like visual takeaway of all this. This is the starch under a microscope. Here's the scale bar up on top. So it's not very high magnification, but you can get a picture of what the starch is actually looking like in these tubers. These are all prepared in the same way. So in the equituber, you have some small starches, not very cluttered, not quite dispersed. Makalitako, even less starches, much more dispersed, small as well. In a small tuber, this is actually a small corm, you can see a lot of the fibers sort of covering the encasing the starch. These are different cells. In shimuko, you see very few starches, but a lot of fiber. This is a panjuko, the one that's related to sweet potato. You can again see packets of starch encased in different cells. A lot more starch in this case, which is why it was probably highly fermentable. And you have sweet potato starch, not so much fiber, but small starches, fairly dispersed. And then you have white potato. And there's almost no fiber that you see, and just the slide is obliterated with starch. So this is all I have, and I have to thank all of the people and the collaborators and the mentors that have made all of this work possible for me, and thank you so much for your attention.